face in the particular case of gene delivery and targeted delivery system is its biodistribution and how it is reaching to the target organ. In case of gene delivery, how it is reaching to the gene, that is intracellular at the nuclear level, it is working. So is it able to reach there specifically without any getting degradation outside its target? So this is the main purpose of knowing this biodistribution and pharmacokinetics. So we know that from the absorption site, wherever we are giving, whether it is, say for example, intramuscular injection we are giving, then it is entering into the blood. From the blood, it is reaching to the different organs of the body. But in this particular one, we are going to reach our target organ, and specifically our target drug delivery system should concentrate. So all these will be taking place from the absorption rate constant to elimination and distribution rate constants. All of them will be present in the pharmacokinetics of our drug delivery system, whatever we are giving. Now the problem comes, how can we estimate what we have given? It is present in the small amounts. During the administration and R&D, they will be most of the times radioactive material, they will be putting it, so that how the small tiny international units of this particular delivery system entering it, easy to access it by the radio tagging. So this is easy to monitor by the imaging techniques. This is the one which can be taken place in the minute quantities in the R&D stage. For the regular runs, how we are going to make it is based on the either its effect in the pharmacokinetic or pharmacokinetic dynamic manner, we are able to see it. So in those cases, we have got so many parameters as applicable to the regular delivery system applicable here also. And we are going to see the characteristics of the drug also to make our delivery systems, so that it will be having an impact on our reaching to the target site. So these are all the things which are necessary to know. And the important consideration which we are going to give is now that, say for example, I have already told you that this is a highly small quantities and sophistication is required. We can explore it 1 or 2 times or 3 times during our validation and R&D. But regularly, whenever using in a regular pharmacokinetics, we cannot do it. So because they are particularly designed to act in a particular site, so we need to know the time profile of the drug, how it is responding. So we can't do these radio tagging, and because the radioactive elements which are going to give in regular manner, it is not possible, and it is not worth, and it is costly and not suggested also. For that purpose only, what we do, in R&D itself, we will do the IVC, that is in vitro in vivo correlations. And these correlation data we will be releasing and seeing that our delivery system reaching or not. It means we are not going to directly estimate our quantity, but we are correlating with some effect or the simultaneous metabolism or enzymes which are released in administration of our delivery system, and what changes are being made by this delivery system introduced. These techniques which will be developed to remove the sample and outside, and they will be tested for its quantities of certain responses which are present, and we are correlating with that to travel how it is made into the particular site. So this is actually what will be done in the case of repeated cases when we want to do it without doing anything touching to internal organ, but we are accessing a tissue or a accessible blood or any exudate, or it is a urine or a sweat such samples, which we can do that also. And also we can do the pharmacodynamics, how the effect is observed after administration of this can also be done. For that purpose, we have got some specific consideration of pharmacokinetics applicable to the only gene therapy and target drug delivery systems. So in the in vivo evaluation of the drug delivery system, there are 2 basic questions. Is desired rate time profile of the release is shown in the in vitro actually obtained in the in vivo? Because we are having a correlation. Is it really having a correlation? Is it going to be worth mentioning, or we want to stick to that by having this particular in vitro and applicable to the in vivo? And the second question is the desired response time profile obtained in vivo? So because it is not having a correlation, can we do directly in vivo? This is the first question which you are going to have. So to answer the first, we have got 4 approaches. One is assay of the unreleased drug or the drug which is present in the outside, not in the target organ. It means if it is present more and more outside the target organ, it means reaching to the drug to the target organ is very less. Assay of the parent drug or the metabolite in the blood, because we are not able to access at the particular organ, we are accessing in the blood. Or the assay of the parent drug or metabolite in the excreta, because even the blood is also painful technique, we can't do it. We can be watching in the traces of the drug or metabolite in the excreta. And even we can't do that one also, because it is minute quantity, we cannot estimate it at all. In blood or a tissue or a target organ, it is not at all possible. Then we are estimating the quantity of the response intensity, how the pharmacological response is appearing, means whether it is a decrease in the tumor size, whether it is having a effects properly which is intended, or it may be a any pharmacological response, or reduction in the BP, or reduction in the sugar, or any response we are intending, actually it is observed or not. We can see that one also. In the second question, if the pharmacological and clinical response can be quantified directly, like the blood pressure and dilations of the people, or intraocular pressure, which can be measured directly, so we can take the pharmacodynamics, like measurement, reduction in the tumor size, reduction in the swelling. Such things we can estimate and see that whether the drug is actually reached or targeted it, or the targeted approach is releasing the drug and acting or not, we can see it. So the selection of the choice and the method of estimation of the input rate and pharmacodynamic effect is very important. What way we need to choose, we need to determine during initial stages itself. Okay. For that purpose, we are having 3 types of evaluations done. One is the steady state analysis, another is pharmacodynamic aspect, another one is the temporal aspect. Temporal aspect is the effects which is something relevant to their time duration. Most of the drug targeting is to deliver the drug at the site of action. So at the same time, and minimize the toxic effects, because we don't want to have them, the drug effects in the outside the target. In the pharmacokinetic terms, its aim is to maximize the ratio of the drug concentration at the site of action than that of the organ where it is having a potential toxicity. So for that purpose, the 3 things which we are going to do it, that is steady state analysis, pharmacodynamics, and temporal analysis. So we have already seen that in the site-specific delivery, we are having a central compartment. It is being given as a drug, and this drug is input to in the form of injection. So that is actually having a carrier or some support pharmaceutical ingredient that is tagged to the drug, and we are giving an input to the central compartment. From the central compartment, it has got opportunity to enter into the other tissue compartments and target organ. So the target organ is present at the top, and other tissue, they are present in the bottom. So the other tissue compartment in the bottom, if it is present, they are resulting into the toxic effects. And the target organ, when the drug is going here extend to the target organ, and it is reaching to the beneficial effects. So this is actually what we are going to do in the target drug delivery. So we are giving to the central compartment by the means of IV directly. If it is not IV, if it is IM, we have got an absorption component, as we have seen in the previous slides. Then we are entering into the central compartment, then it is accessing it. Okay. Sometimes what will happen, if targeting is not there, from the central compartment itself, the drug will get degraded and eliminated. It means the targeting index or the targeting power of the particular delivery system is nil. It is not having. But you will be having highest concentration in the target organ. If you are able to get it, then you will be having a highest targeting index. So then you can say that my drug is targeted precisely. If you are finding in the tissue compartment other than the target, and your toxic effects are very much high, so it is not at all possible. It is not worth to use at all. There is another option which is given in the top, is giving directly to a target organ. So whatever that organ it is present, and we are giving directly injecting to the target organ or target site in the local, like direct injection to that. That is bypassing all the bottom events. This can be done for the gene therapy and both the targeting also. how to study it in the steady state analysis out of 3 and how to do it. So we are having one component is that from the central compartment, we are having S is a central compartment. After reaching the steady state, it is having a rate that is with the target tissue T and the rest of the tissues R. And the rate will be having in a reversible or irreversible manner depending upon the type of drug. And after reaching the steady state, each drug and target will be having some rates. That rate is required to be very high, that is QT is very required to be very high, so that your drug from the central compartment is reaching to the target organ T when compared to rest of remaining tissues R. So the targeting index, that is drug targeting index, can be determined by seeing our drug carrier administration. We have administered with a drug carrier. That means we have got a specific gene therapy or targeting carrier like liposome, or it is quantum dot or anything. And parallelly we are observing without using the carrier direct drug administration and finding the area under curve at the organs of a particular tissue and that is interested, and we are finding the ratio. So this ratio required to be very high, then your target drug targeting index is very high. Of course, here you need to change the R AUC by T by R, which is required to be changed accordingly. You can define. So area under the curve of concentration time profile we are going to see, and this profile, it will be giving the highest drug targeting index, means where the numerator is there with the drug carrier. We are using as a liposome or any delivery system which you are having the highest side at the top, that is in the numerator when compared to the denominator, where we are giving only drug alone without any carrier. Then your targeting index is very high. So if it is 1 means the ratio from the top and bottom is same, means you don't have any targeting. So in the steady state analysis, you don't have any targeting, then you can see the pharmacokinetics is that there is no use of delivering the drug in the carrier because it is not having a beneficial effects by using that particular carrier. The second is the pharmacodynamic aspect, where we are going to have a targeting index. So in this particular case, we are seeing the area under intensity time profile. So in the AUI, we are having the area under intensity time profile is more with the AUI target tissue with a carrier administration, that is a targeting approach. And when compared to the bottom, that is the AIT, that is the area under curve for the drug which we are giving without any carrier, or we are not using any targeting approach. Simple drug solution or drug injection we are giving without any nanoparticle or any carrier or targeting approach or gene delivery approach. So such things which is having targeting index will be very high when target is having a high intensity time profile in the numerator when you are giving a carrier when compared to a simple drug administration. There is a third thing is a temporal aspect. In the time race space, we are giving MI, that is actually maximum intensity. So when compared to your regular, we are making a changes in the DNA and releasing a messenger RNA, and triggering to that messenger RNA, that is a transcription process, we are translating into a protein. This can be done within the cell because we don't want to have a targeting directly injected by bypassing so many things where the protein is having a problem in reaching to the site. This can be done. It means we are using a cellular pathway for the targeting also, and also we are using the cell to have the express our genes outside to get a definite material that is used for the recombinant DNA technology. In the recombinant DNA technology, we are altering the DNA of a bacterial cell and asking the bacteria to synthesize our protein which we are desired, and we are producing it large number in the biotechnological laboratory, and we are purifying them and injecting to the patient. It means one way we are nucleic acid based delivery system we are giving to body of a patient, asking the body of the patient to synthesize the protein within his body. Whereas in the protein based systems, we are asking the bacterial cell to synthesize on the behalf of the body, and outside the body it will synthesize, which is simulated to the human being protein. and it will be injected later into the patient so that he will be having a immediate relief, or it will be acting as a prophylactic purpose. That means curative and prophylactic both are present. So this is the one which we are going to use it. And most of the times there are so many trials which has been made to have this, because the extraction process of the natural resources, if you take it, it is requiring a lot of raw materials and cumbersome steps, approaches to get your material, and yield is very less. That is the reason we are going to entering into that. It is started in the 80s, and there are so many moieties are introduced into the market at present, and only the cellular plant material in this particular case is a yeast bacteria and mammalian cells, and we are using them to synthesize our material of interest. So in this particular case, in the rDNA technology, what we are going to do is, first we are information transfer from the DNA to the rest of the cells by means of mRNA. Then mRNA is going to yield a protein, and that protein is liberated into the blood, or within the cell it is having a functions to do it. Most of the times which we are going to use this phenomena in the target drug delivery, but in the manufacturing of a protein for the curative purposes or prophylactic purposes outside, we are using DNA of a bacteria, what we have already mentioned. So if you take the recombinant DNA insulin, so we know the segment of the insulin which is responsible to produce the sequence of the amino acids, we know it. So we take that mammalian proinsulin, which is having a sequence of nucleic acids. So that mRNA, we know it, in the base we can extract it, and we are converting that into, most of the times that segment is introduced into the plasmid cell of, that is a gene of your bacteria. So the plasmid is a circular DNA material which is present in the bacteria, and we are inserting this segment which is specific to synthesize your insulin, which is having a specific to the human usage, and injecting this recombinant rDNA into the cell of a particular carrier, that is vector. The vector will be growing on its own and synthesize on our behalf. This is actually what the fundamental in the recombinant DNA technology. So what we are doing first, we are doing a molecular cloning. In the cloning, what we are removing a segment of a mammalian mRNA, which is having a specific interest of a enzyme, like a synthesis of insulin or growth factor, or any specific enzyme which is required to be used for the production of particular material or a precursor that will be having affinity or pharmacological action. So that we are making, and we are inserting into the plasmid, and that plasmid is inserted into the vector. That vector is grown, and we are going to produce our protein from the cloned gene. So this is actually what the RNA DNA technology is. Most of the times why we are going to use it for the is MAPS, because whenever we are giving any vaccine, the vaccine is entering into the body and giving a trigger. The trigger will be producing your antibodies. It takes a longer period of the time. During that particular time, the patient may die. So in the MAB, what will happen? We are going to give directly the antibody, which is ready-made available in the pharmacy stores, and asking that particular antibody to attack an antigen, so that we can safeguard the interest of the patient without waiting period of any patient that is making him lethal. So this is actually a MAP technique. We are having so many. In this particular case, previously we are having so many techniques and hybridoma technologies where we have got similar to the human being, but not exactly the replica of the human being insulin or any material. Most of the times we are having these synthesized by making a modifications outside, which is having some errors in it. That is the reason which we are having a problem. But nowadays we have got a recent advancement in that we are going to produce 100% humanized medicines for the injecting into the human body without having any another animal or carrier's effect that is causing a immunity problem, or it is producing a side effect and allergic symptoms. So this is the advancement which we can say it. How we can do this? There is a steps. There are 6 steps are present in this. First, we need to isolate the genetic material from the carrier, whatever we are going to use, and we are using the restriction in them for the digestion, and taking the particular segment, and using the PCR, that is polymerase chain reaction, for the amplification of the particular one, and we are going to produce the ligation of that particular DNA molecule, and the inserting the RNA DNA in the place into the host, and we are isolating the recombinant DNA cells. So most of the time, this restriction enzyme is usually used to prepare the DNA fragment to subsequent molecular cloning, as the procedure allows the fragmented to the DNA and inserting a new block in its place by the ligation. So ligase is the enzyme which is being used, so that we will be able to introduce that particular one. So once we have done it is outside, and we are increasing the quantities, and we are inserting this number of rDNAs into the host, then the host is grown, so that carrier is grown, then we can use its cells for the synthesis of the subsequent production of our any proteins. Most of the times, these ligases and endonucleases, they will be used in this particular purpose, to have the separation from the particular vector and inserting into the same after preparation into the vector. This combination of all will be available ready-made, and we are going to use the same thing in the regular bioreactor itself, the same thing which will be used for all these processes. In this particular case, we have mentioned that it is a PCR process is there. And what is this PCR process? Why are you using in the diagnosis and the production? Most of the times, whatever the DNA which we are collecting is present in the small quantities. So that quantity which we are going to use, and for the patients and outside the body, and we are going to synthesize outside the body. So we are going to use a huge number of the replicas which we are going to use it, because throughout the world patients are waiting. So 1 or 2 vials are not sufficient. We need to produce the lakhs of vials. So this particular thing which we want to make it PCR, that makes the replica of the same thing what we are going to synthesis in large in number, so that we can have the production taking place in the multiple sites, and the number of these organism available to is more, so that we can have them grown in the biotechnological laboratory. So in this particular 3CR process, the first one is the denaturation, annealing, extension, and the 3 steps are present. In the denaturation, as in the DNA replication, the 2-strand DNA helix has to be separated. Unwinding the DNA is taking place. Then in annealing, the primers are bound to the target DNA sequences and initiate the polymerization, so that you will be having a opposite corresponding strand, which is that is prepared into the denatured DNA. That means which has got a helix is got separated. So you will be getting from 1 DNA, you are going to get a 2 DNA moieties. Then extension. The new strands are made using the original strands and as the templates. And again, this process will be keep on going. Only the thing what we require to do is in the media and where we are making this replication, we are going to provide the precursors. These precursors are used to make this particular template multiplied, and we will be getting enough number. After this enough number is there, we will be freezing this particular process and making this DNA inserted into the vector. So these vectors will be synthesizing later. So in PCR, a certain kind of reagent or the primers are used to target the small but a specific part of the virus genome, or any whatever we are going to use as a vector in the question. With the help of this enzyme, the small genomic area is amplified over and over again, so that we will be having the more number of replica is present to use it as a rDNA to introduce into the plasmid. So most of the times we are having in the gene product development, we are having in the molecular biology. First, what we are going to do is the therapeutic gene cloning and transformation and the protein expression is taking place. Then we are going to have, once that cell is made ready, rDNA cell is made ready, then we will be using conditions of the media and temperatures and pH, and we are going to have a highest productivity of that particular rDNA cell, so that it will be having highest protein expression. So the protein is getting expressed, but it is mixed with so much of the bacteriological media and components and precursors and waste products which are coming excrete of the bacteria. Everything is merged. So that is the reason we are going to have protein purification. And once that protein is purified, we need to put it in the formulation where we may be using stabilizers and tonicity adjustments, and then we will be testing them analytically to see that whether it is present correctly or not, and transfer the to the production this technology, so that we will be synthesizing more. This is actually what is each step which is present there. So it is clearly explained here. And the production critical steps are the diagrammatic manner, which is explained later. The same thing which we have already explained previously in the picture diagram we have seen. This is in the lab, and this is in the pilot scale, 10-liter capacity. This is used in the production scale. This is actually removal of your required components. Then we are having a processing and separation of a particular protein, what we are using a size exclusion chromatography, or it may be having a maybe specific substrate addition, that is maybe having a holding, or it may be having a affinity chromatography where the protein is having affinity to your stationary phase. Then all the component of debris is going, then later onwards you can remove your protein of interest. The technique of the separation may be the occlusion or affinity, whatever we are going to use, so that we will be having the separation of the desired protein from the inclusion bodies. The inclusion bodies, most of the times what will happen, you will be having getting added to your process. All of them, it will be appearing as a your contaminants if it is not removed. So we need to use a chromatographic systems. After that, you will be having a end product getting, so testing them, see that whether they are present or not, the purification of the present. So then we are going to use them for the production. So most of the times this gel filtration will also be having, depending upon the viscosity and their affinity, they will be getting also used to separate the specific protein requirement what we have got from the regular mixture of materials. So for this purpose, we are having a water system, which is plenty of waters are required. For that, we are using reverse osmosis, and these plants which will be used for the water injection, which is having a distillation at the end, and we are going to use that water. So these vials, after prepared, we need to make them at their raw material stage or in the final. Most of the times, the protein moieties are heat sensitive materials. So we need to sterilize them in individual raw material and the ampule stage, and using them in the class 100 and clean room services, where we are having these facilities will not introduce any contaminant into our product, so that we will be having, and producting sterilization may not be necessary, where we are taking initial raw material and packaging, they are already sterile, we can get the sterilized product. In this particular case, we are taking ampules or the vials where we are using having a double door entry of autoclaving, where we are initially entering your cleaned vials in one side. In the class 100 side, they will be taken out and going to the filling zone directly by the conveyor belt. This is actually what the conveyor belt which is being shown here. It is having a double slit. One, it is appearing outside it, and other side of your wall, you will be having another slit window that will allow these vials going clean without any contamination later onwards. It will be going in line with your production systems, so that it will be packed and labeled. So This most of the times no man intervention is required. Here it is completely automated system. So the sterility in the class 100 will be possible by this automation, where we are giving the command so that your belt conveyors, how they are moving and taking these vials to the dispensers, where your product is getting dispensed into the vials or your ampules, then subsequently they will be labeled and released into the market. This is completely smooth finish without having any entrapment, sterilizable and disinfectant. If you see this without having a protrusions, and everything is having a smooth finish to easy sterilization and cleaning to achieve your sterility requirements even in the corridors. So most of the times we are having a regulatory aspects. We see that a Schedule Y is pertaining to this of Drug and Cosmetic Act. They will be looking into this, and Department of Biotechnology and the Ministry of Science and Technology of Government and 8th Amendment of Schedule Y. We need to go deeper a little bit. What is the requirement, minimum requirement we need to have it, that is having specific to this particular biotechnological products applicable to this rDNA technology also. So most of the times you will be having how to get this particular specific cloning. Nowadays we are having this CRISPR one, where we are having a CRISPR-Cas9 systems. They are having editing without any problem and simulating to our human insulin or human product, humanized products, which we are going to use. Most of the times this is actually what is a solution for the not having any contamination, because even a simple contamination at one nucleic acid, it will cause the different protein synthesized and yield the product which is not going to meet our site. So how this is having is a CRISPR. Most of the times this CRISPR, what it does is it will introduce a double stranded breaks anywhere where you want to desire it at the guiding nucleic acid that is present. Most of the times this is ribonucleic acid, which is going to guide in a particular sequence, which we can specifically diagnose and synthesize. Later onwards, we will be having a bottom PAM site, that is two double stranded in the DNA. We have got the two sides from the top and bottom, we need to cut it. And in the same place, we can have a ligase enzyme to introduce your specific segment, what you already synthesized from the PCR process, and making that is available so that you can produce the rDNA technology. So this is actually making a foolproof technique to precisely introduce nowadays by using a CRISPR-Cas9 system. And we have got, this is all which is having rDNA technology and gene expression systems. And we have got some more is called as gene therapy. So we have already seen in the we are using most of the times there are 4 different types of vectors which we are going to use, and what are the vectors which are used in the gene therapy, and how we are making these changes in the gene. We want to make it possible within a particular cell of a human body. So for that purpose, we are having a 4 type of the vectors. The first one is the retrovirus. So the retrovirus, they will be highly virulent and they can enter into the any cell. Most of the times, they will be infecting even bacterial cell itself. So these retroviruses create a double-stranded DNA copies from the RNA genome. So the retrovirus goes through the reverse transcription using the reverse transcriptase and the RNA. The double-stranded viral genome integrates into the human genome via the integrate enzyme, and these integrate introduce, that is inserts the gene anywhere because it has no specific site, may cause the insertional mutagenesis. So that is the reason the vectors are derived from the human immunodeficiency virus HIV and are being evaluated for the safety, because the virulent and pathogenic portion in the retrovirus also getting inserted into the gene, then it will cause the, even small traces are present, it will cause the diseased symptoms of the what the vector we are using it. So precisely we need to introduce the retroviral gene segment, which is not having a pathogenic part. We need to remove that. So for this purpose, we have already seen nowadays, which is available CRISPR-Cas9 system. We can specifically remove that HIV pathogenic portion, and the remaining thing is to introduce this virus desired site that can be used. As usual, this virus has got the pathogenicity, and that is more prevalent to enter into the any cell that will be retained without having any pathogenicity. So this is actually what we are going to use. The second part is the adenoviruses. So they are the double-stranded DNA genome that cause the most of the times respiratory and intestinal and eye infections in the humans. So there we are going to use this particular carrier. So the inserted DNA is not incorporated into the genome, completely genome, but not replicated through, has a reinserted when the more and more cells. Well, most of the times it is used, it will be present most of the times in the common cold, and they will be used as a vector for this. It means this one most of the times in the retrovirus, our original genome may be getting affected, but in the adenovirus, we don't get affected with the complete genome. So it is a little bit, we can say it is a safer, because it will not incorporate into our human genome, so it will not make any changes. So, but it is not replicated through to the regular one, because it is specifically particular cells which are present in the respiratory intestinal area of cells. That is the reason it is restricted to those diseases applicable to these organs. It means we have got 3 types of delivery systems and the 3 levels of targeting. That is at the human organ level, and we are having a cellular, that is at a particular cell specific in the organ level, and we have got a nucleus, that is a type 3 nucleus level. So in the retrovirus, we can categorize this carrier to the nucleus level. It means it is directly entering into the site where we want to have a level 3. In the adenovirus, we can have it as a cellular level, we can have it that is at the particular organs which are going to be restricted. So we can use this targeting of this first order targeting, which can be very well used. Another one is the adeno-associated virus. This is a third vector which is used in this particular gene therapy. It is a virus, small and single-stranded DNA. Don't get confused. What is this single-stranded DNA? DNA is always double-stranded only. Why it is present as a single-stranded DNA? Why can't we call this single-stranded as RNA? That is another question you may be having in the mind. So because we are having a RNA virus also. Most of the times RNA virus, and why this single-stranded DNA, what is? Only the one component is, it is a deoxyribose, which is present here. So deoxygenation is not present in the RNA. Because of the deoxygenation, they are more stable when compared to remaining RNA viruses. That means they will be dying very fast, but they will spread very fast and they die very fast also. This is actually, the genome will be very tough to getting affected because of this deoxygenation. Because of that reason, it has got its own advantages and disadvantages also. So this insert the material at a specific point on the chromosome, that is actually here from the parvoviruses family. It causes no known disease when we are using it as a vector, not like a retrovirus, which we are isolating from a particular diseased condition, and we are taking them. So pathogenicity obviously present. And this in the adeno-associated virus, we don't have that pathogenicity. Does not cure and trigger the patient immune response also. and low information capacity. Most of the times their length is very less, so that we will be having a specific segment of a least genetic number we can introduce. Gene is always on, so the protein is always being expressed, possibly even in the instances where it does not need it also. So this is actually what is used for the synthesis of the specific protein which is required, not to shutting down the protein which is causing a damage to our body. So this type of adenovirus, it means we want to introduce this one to have a switch on the gene which is already present off function. So hemophilia treatment, for example, a gene carrying vector can be injected into the muscle and prompting the muscle to produce the factor 9, thus preventing the bleeding. Because in those patients, factor 9 is not present. By introducing this adenovirus, and which is having this carrier, or we are taking this particular adenovirus genome and it is having the factor 9 segment, which is already getting inserted there, so that that factor will be released into the body patient in a particular site, and it will prevent the bleeding. So this is actually what the function of adenovirus carrier. So this was actually the successful, which was already been studied. The fourth one is herpes simplex virus. So this is actually a double-stranded DNA virus that infect the neurons, most of the times, which will be used for the exterior purpose and the nerve cells, and targeting and changing the degeneration of the nerve cells where we are having a disease. Most of the times it will be used. It means we have got a 4 type of virus vectors. One is retrovirus, adeno associated virus, and herpes simplex virus. These 4 vectors which will be used for the different levels of your targeting depending upon our purpose. So for the specifically for a particular organ, we are using the adenovirus, like bacterial infections of or infections of your respiratory tract. For the nerve cells, we are using herpes simplex viruses. For the entire genome sequence and modification in the genome itself, we are using the retrovirus. So this is actually what the specificity we want. Accordingly, we are going to introduce these vectors and choose these vectors for our gene therapy. And in the gene therapy, which part we want to make it? Entire genome, retrovirus is better. Only nerve cells, herpes simplex is better. If you want to use only for the respiratory tract infections and respiratory tract modifications, the gene, which is adenovirus, is better. So this is actually how we are going to choose these vectors and making them to use for the treatment of the diseases by the gene therapy. Another option which we are having is the non-viral vectors. The first 4 viral vectors, it has got one disadvantages. Say if you have got a retrovirus, it is even genome is modified and the person behavior, and he may get the new disease also. Such problems are there. So if you are making any changes, even small segment changes in that particular one, it will produce a wrong sequence of a protein. It may not be functioning. So this entire intervention is waste. So why can't we use the non-viral vector because of these problems? And sometimes you may be having rejections. So why can't we make a non-viral options? So this is actually a non-viral option which is having a direct introduction of the therapeutic DNA directly into the nucleus, that is cell near to the nucleus. So, but this can be possible with only certain tissues. Not possible to the interior tissues like a heart, where you are having a rib cage and you have got endothelium and you have got internal you have got tissues where you need to have the cell reached. It is very tough to reach directly to the DNA of a particular heart. Such things which is not possible. And another problem in this particular non-viral option is requires a lot of DNA, so that we will be reaching there. Even lot of DNA got wasted also. We can have some segments which is possible to target to the particular organ. Most of the times it requires a creation of artificial lipid sphere like a liposome, so that it will be having a bypassing reticular endothelial system. So we can use the non-viral option like a liposome, so that we can bypass the endothelial system, that is RES, reticular endothelial system. So chemically linking the DNA to molecule that will bind to specific receptors, that is ligand based. That we have already seen. We are producing the protein. That protein has got a specific carrier interaction. So the receptor ligand interaction, which will cause the transfer of the drug which is attached to the particular one, or having a RNA introduced taking to the within the cell. So that is the reason directly we are having a DNA engulfing by the cell membrane, and most of the times this will be foreign to our body, so it will be having a less effectiveness if it is having a endothelial system effect and making them to reduce. Trying to introduce a 47th chromosome, most of the times this even exists alongside of the 46. These 46, most of the times we will be having a somatic and sex genes. You know that this is which we are going to have it.